Scientists have created the first synthetic cell. I'm Shelley Banjo with Lee Holtz, senior science writer, The Wall Street Journal. Lee, tell me what's happened here.
Well, this is a threshold moment in the history of biology, scientists tell me. For the first time, researchers have been able to create a synthetic living cell. For 35 years, researchers have been cutting and pasting little bits of gene material to create plants and animals. But this is the first time we've been able to craft an entire organism, doing its entire set of genetic instructions.
So
and this gives us real power over life that we've never had before.
Take us into the chemistry lab over here. I mean, what are people actually doing? How do you create a cell?
Well, actually, what they do is they take computer code, and in a very real way, they're taking computer code and turning it into a life form. They start out with the DNA sequence, which is just a code of A's and T's and G's that represent the chemicals of life. 1 million characters. It's the largest genetic instruction set they've ever worked with. They work it all out on a computer. They rewrite it. They edit it. They actually put literary quotations into it, James Joyce and things, just so it would stand out from a natural organism. They then took that code, mailed it to a DNA synthesizing company in Washington State called Blue Heron, who just in the business of synthesizing DNA. They ran this up on their synthesizers, turned this code into the chemistry of DNA, and gave it back to the researchers, who then used the natural abilities of certain cells, yeast and bacteria, that can stitch this material together. I mean, like, women making a quilt. It's quite remarkable. Wow. Into the whole genome, which for our current capacity in genetic engineering, an immensely long string of DNA. Very brittle stuff, very fragile. They then took that, put it into a cell from which the other DNA had been removed. This new DNA, this new genome, what you call the complete set of genetic instructions, like a new operating system, like a new version of Windows or a new version of Snow Leopard, took over the cell and transformed it into a new species.
So what are the implications here? What could this mean for the scientific field?
Well, this gives us like a fundamental ability to really craft organisms to serve medical and industrial purposes. And this is something scientists have been talking about for 35 years. And, to a small degree, we've been able to do it. I mean, you can get genetically engineered insulin, things like that now. But now here you're talking about being able to sort of take over the entire metabolic pathways of the cell to do things. And I stress, this is very hypothetical and theoretical at this point, but like design a form of algae that can take the carbon out of the air and transform it through its digestive system into a usable fuel. Exxon right now has a $600 million contract with the company that funded these experiments to do just that. It's very far off, very hypothetical. There are at least a half a dozen other firms working in this area, which they call synthetic biology, to try and do these things.
I was going to ask you, who's behind this, and is someone going to make some money off of it?
Well, I assume Craig Ventnor, who is the chief scientist in charge of all this, will stand to profit at some point. He is the fellow who was responsible for the private enterprise effort to sequence the human genome. His company, Synthetic Genomics, put up about $30 million for these experiments, in return for which they get the intellectual property rights.
Well, we're going to have to keep tabs on what happens. Been speaking with Lee Holtz, senior science writer with The Wall Street Journal, and I'm Shelley Banjo in New York with The Wall Street Journal Digital Network.